Do any of you working on a particular gene that you want to learn more about? Who's working on a gene? Yes.
I'm currently working with ZFP right now.
What is it?
ZFP, zinc protein.
CFP.
Yeah, ZFP.
Oh, ZFP. Okay.
I have no idea what it is.
Okay. Is it ZFP? Is that, it's got to have a number, right?
ZFP36L2.
Okay. No, that's perfect. Okay, so we'll do it. So in what context do you want to see that gene? Like in what cancer?
We're looking at pancreatic cancer.
Okay. I think you're in another, is somebody else in your lab looking at NRLP3? Okay. I remember the last question, somebody said pancreatic cancer and they were looking at that thing. So here's what we're going to do. We'll take single data set. And then we got to, so we're going to select our data set. So here, in this case, why are you doing this? In tumor tissue, let's go pancreatic. There we go. Okay, we now have pancreatic. Here's what I want you to do. Again, let's go to our N. Let's sort by highest to lowest. All right. Now what I want to do is I want to find a pancreatic data set that isn't, let's see, this is bulk. This doesn't say RNA. I want RNA. So let's go on the second one, pancreatic ductal adenocarcinoma PDAC. Select that. This is telling you microarray gene expression profiles, 139 pancreatic tumors versus 102 adjacent non-tumor tissue. I like the adjacent tumor. I think it's a better comparison, so let's look at that. So confirm selection. Okay. We will say, here's what I want to do, is I want to, if I want to figure out what a gene's doing in a particular context, let's go to find about halfway up, maybe a little more towards the top, find correlated genes with a single gene. Everybody see that? Click on that. All right, let's go next. Okay, up here, now we see correlate with gene reporter. Tell me your gene again. CFP 36 L2. Okay. As I type that, what will happen is it'll come up here like this. Just click on that. Okay. We can leave all this information, log 2. Again, false discovery rate. Basically what it's saying is you're running a lot of correlations. Do you want us to account for that? I usually just leave the defaults. A false discovery rate of 5% is fine. A positive, negative, we want both. If we want just positive, we could do that as well. But let's hit submit. I think that's the hardest thing to do with this stuff. As far as what I know, it's you've got a gene. How do you figure out like what else it's working with? Like, and if you look here, man, oh geez, like the correlations here. This is going to be a good one, I think. We got our gene of interest up here, and then we've got BRD4, but the correlation R on that is 0.894. There's going to be a lot of like similar genes. This is cool. All right, we'll see how well this works. So here's what I want to do. Just like we did when our class comparisons, we now have, does everybody understand what an R value is? Correlation coefficient. Does anybody not understand one? Positive 1 means perfect correlation, positive correlation, and negative 1 would be perfect negatively correlated. And everything's a measure in between there. So let's take a look. Let's go, just like we did, save selection as text file, no header. You'll see the strip file link. click on that. And now let's save this somewhere. So I'm going to say file, save as, save page as. What was the name? ZFP36L2. Dang. And I'm going to save that. Let me open this. So what we want to do is we want to open it with, again, where's my desktop? Oh, there it is. Open with Excel. Okay, this time let's look at things that are the best friends of your particular gene. So under R value, we're going to do the same thing. Go wherever you have a sort. Sometimes you can right click on that and say sort, blah, blah, blah. But here's what we're going to do. Yeah, sort largest to smallest. Okay. Let's do the same thing we did last time. So what I want you to do is put your cursor at B2. Let's copy the top 200 correlated genes. So 201, mine is TMB IM6. And copy. Now let's go back to string. Again, multiple proteins. Since you've used this before, the Homo sapien should already be there. But let's paste in our gene list. Hit search. Recognizing stuff. And then hit continue. Ooh, cool. So in this instance, as I'm just kind of looking at here, a few things are getting my attention. I got a lot of Rab genes, for sure. But I also have this AKT1 in the middle. RAC alpha series. This is a very important signaling molecule. And I think it phosphorylates a bunch of stuff. So in this instance, does anybody see where our gene is? ZF, where's our gene? ZNF, I see a ZNF here. Maybe see it if you see it, tell me if it's down, left, right. Where's our gene of interest? ZNF, there's another ZNF. All right, well, if somebody sees it, let me know. And that way, what we could do is if we find our gene of interest, then we'll say, hey, in this thing, what is it connected to? And is it known to be connected to that? Oh, here it is, right? ZNF 3. Okay. Oh, here? Wait a minute. This one? There it is. Here's what's interesting to me is that based on just literature and just, in the database, it's only connecting to 2 of these genes on here. But in actuality, I think it's involved in all of this stuff. Remember, especially if a gene hasn't been studied very much, the amount of information known about it is probably very, very small. So again, here's our gene. This is probably its gang. Probably got a captain over here and another captain over here. I would definitely, and they both connect to each other. Let's figure out what these genes do. So if I go down to analysis, right? I look at biological processes. I can sort by false discovery. Containing metabolic, cellular, a lot of metabolic stuff. Macromolecule biosynthesis, nitrogen, primary metabolic process. This is definitely biosynthesis of some kind. And again, usually, so this pathway is probably producing something that's getting modified. You also see Golgi vessel transport as well. But yeah, there's something metabolic going on here. Which makes sense. If a cell divides, you're typically going to speed up your metabolic process. So, I mean, this might be something we can go down here. Molecular function, most of these are, transcription co-regulator activity. So I would say a lot of these, we can color that, are involved in transcription. I'm also going to color the metabolic process. Positive regulation of macro-metabolic process. And then where the hell are these things at? These are bound somewhere in a membrane-bound organelle. Intracellular. So maybe these are in the mitochondria. That's what's kind of making me think maybe mitochondria. I'm not sure. But they also have the nucleus. Maybe these are in the nucleus. So we go down. We've got vasopressin regulated water resorption. That's interesting. But now we're also seeing some specific reactome pathways. We're seeing, go more. Heme signaling, there's that mitochondrial, but we're also getting Rab signaling. So I would say whatever this gene is involved in is probably associated also with, I think Rab signaling is probably playing a big role in this as well. Tissue, expression, where it's at. These are certain phenotypes that might be associated with these genes. The p-value isn't, the false discovery rate isn't that great. Lipoprotein, maybe this is lipoprotein metabolism. And then anything else. And a lot of these are associated with phosphoproteins. So genes that phosphorylate other proteins. Come on, work you down. Ooh, is that P10? So again, this would be my starting point. This is what I would use to kind of figure out what's going on. I would also start looking up maybe more, what does AKT1 do, right? Since it seems to be interacting with a lot of stuff, is this somehow associated with all this? Include processes including metabolism, proliferation, cell survival, growth, and angiogenesis. That's like all the, like if you're a criminal in cancer, these are all the things you want to be able to do, right? I want cell survival, I want to proliferate, I'm going to like rev up, we're going to go really fast with metabolism, and then we're going to have a lot of blood vessels to give us food, right? Actually, AKT1 is a very big gene in cancer, so for sure. So, but again, do that, you find correlated genes, you can basically take those genes, put it into this program, and get some pathway analysis.